Okay, so that wonderful subject out of the way. Let's get started today on virus structure. We're going to talk about actually basically all of these various different models of virus structure, and I should really say virion structure here because, of course, that's only part of the virus life cycle. Got icosahedral, some combinations of icosahedral and helical. One of my favorite viruses up there in the middle that we'll talk a lot more about later, partly because I discovered it. And then what we won't talk about is the structure of this virus up here because we actually don't know what the real molecular basis is for that particular structure. But first we'll talk a little bit about what we talked about last time, which is really one-step growth curves. And this reminds me that there are a couple of postings on D2L about one-step growth curves. So if you're interested in sort of how people came up with these ideas, what the process was that was involved, all of those papers are posted on D2L. There's a whole one-step growth curve folder on there. But basically, it's about detection and counting of viruses, which, let's say, kind of the gold standard is the plaque assay. And so growing cells, a vast majority of host cells, very small amount of virus relative to that. You do an infection, and then those infections spread across a plate. So you have multiple rounds of replication that take place in these plaque assays, and then you have to have some way of detecting what that plaque is. Now, in the case of bacteriophage, it's great because they eat the bacteria, and so you can literally see a little hole. And it turns out the people in the recombinant lab, those of you who are wondering if you actually got plaques, yes, you did. So that actually worked. One-step growth curve, the whole idea really of the one-step growth curve is to try and do a simultaneous infection. And so you can follow lots of viruses replicating at the same time in a more or less synchronous fashion. So you don't have to try and look at a single molecule, a single virion. You're looking at a whole population at the same time. And so the way to do that is to have very high multiplicities of connection. So again, lots of virus particles per cell that they're going to be infecting, and that's all due to the Poisson distribution. Going back to this one-step growth curve, basically all that you're doing over that one-step growth curve is measuring over time what's happening to your population of various different viruses. So you can do this as a plaque assay. You could be looking at proteins. You could be looking at DNA, whatever process you're looking at. But just looking at these one-step growth curves, you can get a lot of information, whether it's likely to be a enveloped virus or a non-enveloped virus, whether it's a one which is producing very large numbers of virus particles per infected cell or not. So you can get really large amounts of information from these one-step growth curves. And they're pretty standard. The main problem with the one-step growth curve is you have to have a pretty high MOI in order to have this nice one-step growth curve, have nice synchronous infection that's taking place. We talked a little bit about some of the virus replication cycles, and again, this is really an overview. If you think about the virion on the outside, it's got to attach to the cell, it's got to get inside the cell. Then you have early genes being replicated, and those are usually the ones that are important for getting the virus to replicate. Once the virus is replicated, then you've got the late genes, which are usually important for putting together the virion. And so they can actually escape, which of course is another process to move on to the whole virus life cycle or replication cycle as we have here. Now, that process, again, is going to be different for every different virus, and so we'll be going through all of those processes. One of the things that's pretty common in all viruses, and this is the Baltimore class, is just a different classification. We may get to taxonomy at the end of today's lecture, we'll see. But basically it's all about the messenger RNA. So how you get to that positive stranded messenger RNA, which is what's coding for your proteins. You can have single-stranded RNA viruses, which have a positive strand, so you're good to go. Doesn't really matter, genome comes in, no problem. Double-stranded DNA viruses where you have the normal cellular machinery, which can make messenger RNA, so those are pretty easy. The weird ones are really the negative strand RNA viruses that then have to make a positive strand in order to make their proteins. So they have to have a polymerase that comes with them. So they have to have that protein, that activity, because cells don't have at least not processive RNA-dependent RNA polymerases. Then you've got the single-stranded DNA viruses, which are also very bizarre because the cell can't deal with them. They've got to make a double strand of that DNA before they can make messenger RNA. And then you've got the retroviruses, which are even one step beyond that. They have a positive strand RNA genome, but they go through this DNA for single-stranded and then double-stranded intermediate. So those are the main classes of viruses that are out there. There are questions about last time. I didn't put in clicker questions, so if people have to take off, you can do that now. But I'll have a clicker question a bit later on in some of the virus structure. And next week, those clickers will actually be counting. So let's move on and talk about virus structure. Again, it's really an outline. One of my favorite ones here. This is so pretty. We'll talk about some of the methods, how you go about determining these kinds of structures. Real important concept is icosahedral symmetry, and so that's what I've got my various models up here about icosahedral symmetry. Yeah, may look like an uninflated soccer ball, but it's really a nice model for understanding icosahedral symmetry. One of the really nice ways to define virus particles due to icosahedral symmetry is this concept of T-numbers. And so we'll talk about the triangulation number and give you a number of examples of that. Then we can talk a little bit about virus envelopes and assembly. We actually know a lot less about these guys down here at the bottom. And then, as I mentioned, if we get to virus taxonomy at the end, we'll talk about that too. Key concepts for this lecture, really, it all has to do with simplicity. In most cases, viruses want to be as small and simple as possible. We'll look at some exceptions to that rule as we move along. But basically, the simpler you can do it, it seems that viruses have evolved to use those very, very simple patterns. So as easy as possible. And so the way to do that is to have very few proteins fitting together in very symmetrical ways. And so that's helical symmetry, which is actually probably the most regular way and easiest way to package that nucleic acid, which is inside the bag. Turns out it's got problems because it's got ends. And ends are a real problem. Hopefully you remember from that other class that will remain nameless. Ends are something which is really problematic, particularly for DNA. Think about telomeres, etc. Icosahedral symmetry solves that problem because it's spherical. But the big problem with an icosahedron is it's relatively small. And then you can't package that large a piece of nucleic acid. So there are constraints on the size of the bag. And then when we talk about taxonomy, it's really about, should be as far as I'm concerned, about genomes and structure. Turns out that most of what's done now for taxonomy doesn't consider those very much. So virion structure, again, as mentioned before, generally the simplest structure with the smallest number of proteins. And the reason, again, for that, for most viruses, smaller is better, because you just think about how viruses are undergoing their replication. It's not 1 to 2 to 4 to 8 to 16. It's 1 to 0 to many. And so the process there is many, lots of particles reproducing very quickly. And so to do that, you have to have relatively small, relatively simple kinds of capsids. And so we'll talk about that process here in much more detail. There's what people call the viral capsid paradox, and that actually I think is a really interesting one to think about, because if you think about, again, our bag of nucleic acid here, it's got to be really resistant to the external environment, because it's got to be able to encapsulate and basically protect that nucleic acid from the environment. The extreme case we work on viruses that are functioning in boiling acid mud pots. So, clearly there's this protective aspect that you have to have. But at the same time, all of these viruses have to replicate inside a cell. So you've got to have this really tough capsid on the outside, but as soon as it gets to the right kind of cell, it's got to release the genome so it can go ahead and do its thing. So there's really this push and pull between, having something that's really rigid and stable and something which can be very easy to open. And so we'll talk about some of the ways that various viruses have dealt with this issue. Icosahedral symmetry, again, is a really important concept. Again, the idea here is it's getting close to a sphere. Spheres are a problem because they're round, and it's hard to get your individual proteins to put those together to make round particles. So icosahedra are about as close as you get. And the way to deal with that problem, that they're relatively small, is this process called quasiequivalence, whereby you don't have the proteins fitting together in exactly the same way, but they're fitting together in almost exactly the same way. And we'll look at that in much more detail as we move along here. How do we know about viruses? I should say virion structures. All depends on really big, expensive pieces of equipment. This is the Techni F20 transmission electron microscope, which is in the basement of Science Building 1. So in fact, we use this quite a bit. I don't think that that image of the SSV particles that we had there is actually from this microscope, but at least those people in the molecular biology, say recombinant DNA lab, they may even get a chance to use this microscope a little bit later on. This is the sort of next generation of that microscope over there. That one, by the way, cost $1 million. I think this one's about $4 to $5 million. But this one, particularly here, now one of the really cool things, I don't know if you guys know, probably the #1 electron microscope manufacturer in the world is in Hillsboro. So a place called FEI, Field Emitters Incorporated originally. So FEI Corporation, which you can hard to see here. But they, in fact, built both of these microscopes, and we have quite a nice collaboration going on with them. The really neat thing about this particular microscope is it does everything at very, very low temperatures. That's what the cryos is all about here. And so that's really critical for doing cryo-electron microscopy and particularly image reconstruction. So we'll look at some of those images a little bit later on. But with this microscope over on this side, what we do is mostly what's just called negative stain. And so these are various different virions here that have been prepared with a negative staining protocol. And the whole idea of a negative stain is basically what you're looking at is a electron-dense material. Usually it's some kind of heavy metal. We use uranyl acetate. A lot of people will use phosphotungstate. But basically, it stains everything except for your virion. So here's a nice example of a bacteriophage with an icosahedral head and a nice helical tail. This is one of my favorite negative stain electron micrographs. This is from an influenza-type virus where you can actually see the genome particles inside. Very rare that you can see something like this. This is a pox-like virus. This is an adenovirus, and that's a filamentous bacteriophage up there at the top. So negative stain is great because it's really quick, it's really easy to do, and you can run through quite a lot of samples in the process. The problem is that you're, only looking at what's not there, which is the negative stain aspect, but also you don't get a really high resolution out of this process. And in many cases, this incubating it together with the stain and also putting it at the very high vacuum that you have to use inside these electron microscopes messes up the structure. So those are some issues with negative stain. There are a number of other techniques. Positive staining, and this is an example here of HIV budding off of a cell. This is instead of just taking your virus particles, mixing them together with stain, putting them on a grid, which is what we do for our negative staining, here is a much more involved process until you get here. But it gives you information about the cells and the viruses that are associated with them. And if you're lucky, and there's a lot of luck involved in this process, you can end up finding things like this where the cells budding off a virus, and you can see that budding process. So if we're looking at cell-virus interactions, thin section is a really nice way to go. These really pretty pictures that I'm showing, and we'll show a whole bunch more of today, really mostly come from those really expensive microscopes. So cryo-electron microscopy, usually single particle reconstruction, which we'll talk about, I think, on the next slide, if I remember correctly, or X-ray crystallography. Now, cryo-electron microscopy gives you much higher resolution than that straight negative stain process. The cryo-electron microscopy is really nice because you're imaging in ice, which is frozen really, really fast, so it's non-crystalline ice, it's a vitreous ice, all kinds of interesting ways to do that. Be that as it may, it doesn't change the particles very much. It seems to do very, very minimal kinds of change in the particles. So you can image actually really pretty big particles in this process. On the other hand, if you're really lucky and you can get regular crystals that form of your virus particles, you can actually get high-resolution, atomic resolution structures from a crystal of a virus. But it's really, really hard to crystallize viruses. And that's the main problem that you have there. So let's look at single-particle reconstruction. This is sort of your classic cryo-electron microscopy process. That's what we've got up here at the top, the red blob. You take a picture, and that picture will have many, many, many of these virions in it in lots of different orientations. And then the key is to find all of these virions, get rid of all the crud, which is in here. And anyone who's done electron microscopy knows there's always a lot of crud in your electron microscope preparation. And then line all of these particles up in the same orientation. And then from multiple single particles, reconstruct this particle, the particles that you started with. So there are a couple of things that have to happen for that. One, this has to be really homogeneous, because if it's not homogeneous, when you try and average everything together, it becomes just one big blur. So that's one thing that you have to have. The second thing, and this is always a problem, this is a two-dimensional image that you get out of here. But they're three-dimensional particles. So the calculation process that you have to do going from these 2 dimensions to 3 dimensions is mathematically complicated, particularly if you don't have something which has nice symmetry to it. If you've got nice icosahedral symmetry, it's actually relatively easy to do this step. If, on the other hand, something like this, which has got these projecting pieces coming out in different places, that makes it a lot harder to actually do that process. But this is probably, I would say, the most common, really sort of cutting-edge way of doing virion structures these days, is this cryo-electron microscopy single particle reconstruction. Just as a sort of an aside here, some of the structures that we are looking at, particularly that STIV structure I showed you right at the beginning, we'll talk about it again at the end, that was from, I think, about 13,000 of these individual pictures that were reconstructed to make that image. Yeah?
And there are computer programs that do all that for you?
Yes. There are computer programs that will do that. The big problem is getting a nice preparation at the beginning. And then for a long time, it used to be a manual going through and picking these things out. So picking out 13,000 of them takes a really long time in a very dark room, staring at all these things. There are now computer programs. Image analysis has gotten way better, kind of scarily good if you think about some of the Google and Facebook and those kinds of things. So they're actually using some of that exact same technology to characterize and put together these particles, show that they're the same kinds of things. And it's kind of an iterative process. So if you happen to have collected some of this junk in here that's wrong, and then try and average it, clearly you're going to have a problem. And so you've got to go through and analyze and figure out, okay, this is a reasonable one, this is not a reasonable one. So yes, but there are computer programs to do this, and they're continually being developed. The same thing is true for, oops, not moving here. Let's go forward, see if that'll help. There we go. X-ray crystallography. This is hopefully, you guys sort of more or less familiar with X-ray crystallography? Yes, no, kind of, maybe, head shaking. So the real problem here is really getting your crystal in the first place. Once you get a crystal of your virus, your protein, you name it, the rest of this process is, as you're mentioning, computer programs are basically really good for doing this. So shine X-rays on a regular array, and the real key here is that everything is lined up in a crystallographic lattice. So you've got multiple copies of exactly the same thing in exactly the same orientation. And so that will give you diffraction patterns, which you can then use to reconstruct where everything is. And then from there, calculate your actual protein model. But again, the big problem here is making the crystals, and it's very challenging to get crystals of whole virions. One of the ways that people have been dealing with this is they've actually now made lots of crystals of the individual capsid proteins, and you can actually get usually pretty good structures of the individual capsid proteins. And then take those individual capsid protein structures and fit them into a cryo-electron micrograph where it's not as high resolution. And so this is what people call pseudo-atomic models, because you've got these individual pieces that you put together and hopefully end up getting pretty close to the real thing. And then one of the great things about having a model like that is now you can go back and do experiments to test what you predicted from actually finding out that model. So those are the methods, and this is how we know about a number of virion structures. It's still a relatively small number. But what we found is a couple of, not sure why this is deciding not to work for me here, pretty general principles. And again, smaller is better in this case. We know for a number of small virions that they self-assemble, and when I say self-assemble, basically you can take the individual components, separate them from each other, throw them together in a test tube, and they will make, in some cases, even an infectious virion, which is totally cool. But that process of self-assembly is one, only happens for most of these small viruses generally, but it really allows you to understand and piece apart all of the processes that are going on there. Why is smaller better? Basically, because nucleic acid is just too darn big. You think about the genetic code, 3 nucleotides for 1 amino acid. And nucleotides are relatively big. So anytime that you're trying to make that bag around your nucleic acid, the bag's going to be protein, sometimes membrane, but pretty much always protein with a membrane around the outside. So many cases, in fact, this is said a third of a genome of small viruses is capsid proteins. Sometimes even as much as about half of the genome of some small virus is the capsid protein gene. So it requires a huge amount of that material, and that's just for one protein gene, let alone all the proteins that you would need to make a whole capsid. So the idea here is use the same capsid protein many, many, many times. So that can happen either through a helical symmetric, or helically symmetric, I should say, assembly, which is basically where each of these individual commas, which represents one capsid protein, is interacting with all of its neighbors in exactly the same way. This guy interacts with this guy this way, this guy interacts with this guy that way, etc. And all the way around, if you can wrap this up, you end up with a roll. And so all of these proteins are interacting with each other in the same way. And so you can have one protein that can interact with all of these other ones. Again, helical symmetry is great because it really is an equivalent interaction between each of these protein subunits. The big problem is, what do you do at the ends? So that's... and we'll talk about how this happens for various different virions a little bit later on. On the other hand, of course, the best way to deal with ends is to not have any. And that is the icosahedral symmetry, or the icosahedral structure that you find in many, many, many virions. The whole idea of something with icosahedral symmetry, it's got really 3 different axes of symmetry. A 5-fold axis, a 3-fold axis, and a 2-fold axis, which is why I brought my soccer ball with me here. If you look at the 5-fold axis, basically all that a 5-fold axis of symmetry means is that you can turn it 72 degrees each time, because you get 360, and you end up with exactly the same thing. If you now have a 3-fold axis of symmetry, which is what you have here in the hexagonal face, Now you turn it 120 degrees. You get exactly the same thing back. Then you also have a twofold axis of symmetry, which would be here, where you now turn it 180 degrees, and you get the same thing back. So it's that combination of a twofold, threefold, and fivefold axis of symmetry is where you get your icosahedral symmetry from. Why icosahedral symmetry? In theory, you could also use a tetrahedron or a cube to do that. Turns out that they're probably too small to encapsulate your nucleic acid, plus the corners that are actually formed on tetrahedron, which is just 4 sides, or a cube being 6 sides, are probably too sharp to actually be able to put together really nicely with individual protein subunits. So icosahedra, or let's say a particle with icosahedral symmetry, seems to be the way to go. And again, this is what we find in lots and lots of different virions. Again, it's got 5 folds, rotation axes, 3 folds, and 2 folds. One thing to think about here, this goes back to Euler's theorem from, I think, the 1600s, maybe it was the 1500s, who basically said if you have a flat sheet of anything, in order to fold that up into a closed geometric shape, you need to have 12 of these 5-fold rotational axes. And if you count up the number of pentagons that you have on a soccer ball, you'll find that it's exactly 12. And then you can back calculate the rest of these. But if you think about it, and this is now, again, these comma-like subunits, which are interacting in very similar ways with each other, again, a lot like that helical symmetry that we talked about, you end up with 3 of these proteins for each of the 20 faces that you have in an icosahedron. So a regular icosahedron has 20 faces. Anybody who used to play like war games or fantasy games, way back when, 20-sided dice, icosahedra. So giving away some of my youth here. But those 3 capsid subunits per triangular face gives you 60 capsid subunits. So the simplest icosahedron that you can make is going to have 60 copies of that capsid protein on the outside. And as we'll see, there are a few viruses that can do that. And this is also known as a T=1 symmetric particle. Icosahedral symmetry does not necessarily mean an icosahedron. And we'll get back to that and talk about it a little bit more. This whole concept of quasiequivalence that we're going to be moving on to a little bit later. So you can have icosahedrally symmetric, but not an icosahedron. This is an icosahedron because now we've got 20 equivalent sides to each other, 60 of these capsid subunits that are interacting with each other in exactly the same way. There's a really nice discussion of icosahedral symmetry, particularly for virion particles, at the ViperDB at Scripps, and I just gave you the link, which is there. Let's look at an actual example of one of these T=1 regular icosahedral capsids. This is the Cowpea Mosaic virus. This is one of those very few that we actually do have an X-ray crystal structure for. That's shown down here at the bottom for one of the 5-fold axes of symmetry. If you look at the cryo-electron micrograph here in A, each of these guys is your 5-fold axis of symmetry, nice 5-pointed star. You can also see that quite nicely if you label this with gold particles, which are these big yellowish blobs here. You can also see the nice 5-fold axis of symmetry here, 5-fold axis of symmetry there. The inside is actually the RNA, which is being packaged on the inside of this particular virion. So that's great, but it turns out this only works for relatively small nucleic acids. And in fact, in the case of Cowpea Mosaic virus, I think it's about 3,000 nucleotides. 3,000 nucleotides is pretty darn small. So how do you deal with a bigger genome? Well, basically, all that you do is you put a 6-fold axis of symmetry between each of your 5-fold axes of symmetry. So this soccer ball here is not a perfect icosahedron because we've got our 5-fold axis of symmetry, but in between them, we've got all these hexagons. And so this is, in fact, a really nice example of what we call a T equals 3 icosahedrally symmetric particle. And this is that example here with those individual protein subunits drawn out on it. We've got a 5-fold axis of symmetry here, a 5-fold axis of symmetry here, but between them are these 6-fold axes of symmetry as far as the individual proteins are concerned. And so I've actually drawn them up on this soccer ball so you guys can come take a look at it a little bit later on. So that's the process. Regular icosahedron here, our T equals 1. This one with hexagons between the axes of symmetry, which is a T equals 3. How do you get these T numbers? What's he all muttering about all these T numbers? Well, it's a triangulation number, and again, this comes from Euler's theorem. And all that it is a nice way to describe how you fit those basically hexagons into your icosahedrally symmetrical structure. So this is the process. Your T, which is your T number, is h squared plus hk plus k squared. Now, what the heck's h and k? Well, these are just a vector getting from one 5-fold axis to the next 5-fold axis of symmetry over any of your hexagonal symmetries which happen in between. So we'll start out with the really easy version right here. This is the T equals 1 icosahedra in A. Get from a 5-fold axis of symmetry to a 5-fold axis of symmetry. One vector, one direction. That would be an H is equal to 1. No vector in any other direction. Your K is equal to 0. So T is equal to 1 squared plus 1 times 0 plus 0 squared is 1. Okay, that's pretty straightforward. Now, let's move to a slightly more difficult one. This is now our T equals 3 icosahedra, the soccer ball equivalent, also known as the buckyball. So you may have heard about these. Icosahedra symmetric. Buckminster-Fullerene is another way of thinking about it. So you start out here at a 5-fold axis of symmetry. To get to this other 5-fold axis of symmetry, you need to make one hop here. But if you continue with this vector, you're going to keep going this way. And so you're not going to get to another 5-fold axis of symmetry. So you need to go 1 in this direction, then change direction, and go 1 there. So in this case, your h is 1 and your k is 1. So 1 squared plus 1 times 1 plus 1 squared is a T equals 3. Here's an example. Up at the top here, doesn't have the whole virion on it. But now we have put our hexagons directly between the pentameric subunits. Here, to get from one 5-fold axis of symmetry to the next 5-fold axis of symmetry, you go 1, but you keep going in the same direction, giving you 2. So your h is 2, your k is 0. So your T number is what? 2 squared, which is 4. So that's a T equals 4 icosahedron. Let's take a look at a real example. So here is Cowpea chlorotic Mosaic virus, T equals 3 virus particle. Here are our 5-fold axis of symmetry. There's one right here. There's one up there. There's one here. There's one down there. Getting from one 5-fold to the next 5-fold via the 6-folds, K of 1, H of 1. Clear? Everybody happy with this? Yes? No? Nodding heads? Good? Okay. How do you get to 31 from this one? So I'm going to now, let's see, I think if I give myself, I forget. I used to be able to change the cursor on here. Here we go. So do the marker of a pen. So let's start at one of these 5-fold axis of symmetry. It's a lot easier to see over here. So now we go 1, 2, 3, 4, 5. In one direction, and then change direction, we go 1. So that's 5 squared plus 5 times 1 plus 1 squared is a T equals 31 particle. So this, interestingly enough, was actually the very first T equals 31 icosahedrally symmetric virus ever to be found. So I was, quite proud of that. one thing that is true, because these things are symmetrical, it doesn't matter which way you go from one to another. So we can actually take a completely different approach to doing this. And we'll do the red way. So we'll now go from this five-fold axis of symmetry to this one down here. So now we go 1, 2, 3, 4, 5, and go across 1. Sorry if I'm in the way on that. So yeah, any way you go from one to the next. Yeah. It doesn't matter. So, because again, they're symmetrical. So it could be H is 5 or K is 5. Now, one's going to be 5, one's going to be 1. Yeah.
What's the first one you did? Was that 6?
So 5 in one direction. So here, okay, we'll try again. Start over here. So we go 1, 2, 3, 4, 5, and that's H or K, and then you change and go 1. Does that make sense? Good. There's another question back here?
So the secondary one will always be 1.
In this case. There are cases where that's not true. We already looked at the t equals 4, where it's 0. And you can also have, there are some which are going to be 2 and 2, which gives you a, that will give you a t number of 16. So yeah, there are lots of different possibilities. Or 12, actually, t is 12 in that case. Yeah?
So do you always start on the five-fold?
Yes, you have to start in five-fold. So you go from five-fold to five-fold.
Okay. And it doesn't matter which folds are in between the two five-folds?
They're always going to be 6 folds. So it's going to be your 6s, all the hexagons you put in between your pentagons. Yeah.
I'm confused about the fold. So
Yeah.
the fold, is it just like that? It makes kind of like that five-sided shape.
Right. Yeah. So when I say that's your axis of symmetry. So five-fold axis of symmetry. That's where you can turn it 72 degrees and get the same thing back. But you can also think about that as where your pentagons are going to be. Okay, so this, in fact, is probably, at least for icosahedral particles, 95% are going to be this particular way. Now, they're not going to be t equals 31. They're going to be all different ways of doing this. And sort of the idea of this t equals 31 particle is to show you there are lots of different ways. It's not always going to be an H or a K of 1. It can be multiple different ones. You can make really big things doing this. It's from Expo Center in Montreal. I forget, I think that's actually a t equals 70-something icosahedral particle. But you just the real key here is you've got to find out where the five-fold axes of symmetry are. And that actually is a little bit complicated in one of these really large ones here. Buckminster-Fullerene here in C. There are a couple of variations on this theme which are really kind of interesting. We'll talk a little bit about the Phi 29 virus. It's basically a regular t equals 4 icosahedron where the ends have been popped off, and then another layer of hexagons has been added. This now is no longer quasi-symmetric. It doesn't have icosahedral symmetry. Now it's got a two-fold axis of symmetry here, but you don't have these multiple five-fold axes of symmetry. You don't have 12 five-fold axes of symmetry on it. This is the current model for the HIV nucleocapsid on the inside. It seems to be basically 2 icosahedra with different T numbers at either end, with this set of basically helical arrangement between the two ends. And we think that our virus actually, the one that we work with, oh yeah, and there's the soccer ball, which is that as well. So how do you fit these things together? How do you get an icosahedral structure? Turns out there are lots of different ways of doing it, and we only really know the details for those virions that we have X-ray crystal structures for either the whole virus or the individual capsid proteins that go together. This is the case for polio. Polio has actually 3 different proteins that go together to make its capsid, but they're arranged in a very nice quasi-symmetric arrangement. The genome is just up there at the top. But basically, there's VP1, VP2, and VP3. Each of them have a core structure which is basically identical. And this is what's called a jelly roll structure with a number of beta strands, basically these beta strands wrapping around a core structure. Each of these trapezoids fits together nicely into one of those icosahedral faces. And you end up with, and this is in fact a T equals 3 icosahedrally symmetric particle. It's not completely quasi-equivalent. When you talk about quasi-equivalence, it's actually exactly the same proteins that are being used to put together the virus particle. Here it's 3 different proteins. So a lot of times you'll see a P in front of the T number. That's a pseudo T equals. And that pseudo means you've got different proteins that are coming together to give you that icosahedrally symmetric structure. But these are, in fact, now the crystal structures of each of those individual proteins, VP1, VP2, and VP3. What's in color is this trapezoidal box that basically fits together. If you look now at the cryo-EM structure, actually this is a crystal structure because they've crystallized the Polio virion very small. You can get lots and lots of it, it's relatively easy. Getting from here to here, there's clearly this. It looks more like a 3-fold axis of symmetry here, but that's your hexagonally symmetric point. So from here to here is 1. From there to there is 1 more. So it's a T equals 3. There are, of course, some exceptions to this rule, and one of my favorites is SV40. Only has 1 coat protein. Nice icosahedrally symmetric particle, which is this particle right here. But instead of having the regular number of capsid proteins around the outside, it turns out to have 60 fewer than you expect it to have. And the reason for that is that instead of having hexamers, so 6 copies of each of the proteins at the hexagons you can be putting between each of your 5-fold axis of symmetry, it turns out that SV40 can get away with a pentamer of proteins in basically a hexameric whole, which is really bizarre. And it kind of goes against everything that I learned about when I talked about protein chemistry. It says proteins have 1 structure, or maybe 1 or 2 structures. Well, this particular protein has 6 different structures that can form. All 1 protein. No change in the amino acid sequence whatsoever. Fits together. Interestingly enough, it also has one of these beta barrel structures right in the middle of the protein. And so the beta barrel structure is a very, very common form that you find in icosahedrally symmetric virus particles. And so we'll talk about that in more detail as we move along here. But basically, this, if you did the back of the envelope calculation, I forgot to mention this before, but one really nice way of figuring out how many capsid proteins you have in your icosahedrally symmetric structure, as long as it has regular quasi-equivalency, you just take the T number and multiply it by 60, and you get the exact number of capsid proteins that you have. You remember the T equals 1 particle that we talked about right at the beginning? It's got, just pentamers. How many do you need? How many pentamers do you need? You need 12 pentamers. So to get you need 12 of your pentagons to make your icosahedron. So 5 times 12 is 60, or you can think about the 20 faces, 3 each in those faces, so 60. The great thing about this triangulation number is you can just then multiply that out. So that's great, except if you come to something like this, where you get a T equals 6. Well, how do you get a T equals 6 from h squared plus hk plus k squared? Not an integer. So you have clearly problems. So that also was a really nice indication in this case that the structure was kind of weird. We didn't have this nice quasi-equivalence here. Yeah?
My question was, with 6 potential structures, how is there homology when assembling such a structure if there are 6 potential outcomes of the assembly?
I'm not sure I completely follow your question. I'm going to try and paraphrase it. But basically, how does the virus know what structure it has to be in any of those individual places, and how does this get assembled?
Exactly.
Yeah. So what's that assembly process? We actually don't know in the case of SV40 how it gets assembled. We know that in the final particle it looks like this. But how you end up with these individual things, what seems to be happening, it's actually not known for SV40 but some other viruses, is that you form these pentamers to start with. This is actually completely symmetric. All the alpha form are completely symmetric, whereas all of these are slightly different. So it appears that this forms first, and then that seems to stimulate the formation of these other ones between them.
So there isn't structural symmetry throughout?
There's not a structural symmetry throughout that process. Yeah. Mm-hmm.
So is the T number still 6? It's actually 6 even though there's no.
So this is, it's the 360. There's actually not a good T number for this. That's what the question mark is about. So it's not a T equals 6, because you could, if I said, okay, you got 360 subunits, what's the T number of this? Well, you say, okay, well, I remember what he said. He said, take your T number multiplied by 60, you get the number of subunits. Okay, well, I'll take this, 360 divided by, wait a minute, 60 gives us 6, but how do we get a T number of 6? That doesn't work. So a certain number of allowed T numbers, which may be getting back to your question about, these, whether they're ones or not. So let's take a look at another example here. This is Adenovirus, large, naked virus, no envelope on the outside, has different proteins that are present at the 5-fold axes of symmetry. You can see these nice fibers that project here, sort of the classic Sputnik-like virus particle, Virion, with these fibers. They're also called depentons. Makes it easy to figure out what's going on. I give you one guess what the things in between are called. Hexons. So you've got hexons and pentons. So this is clearly going to be a pseudo-symmetrical structure, but it's arranged really nicely icosahedrally. And so this does have a nice regular T number. And now the T is 25. We can now look at that really quickly here. Here's a 5-fold axis of symmetry. Oops. Except I didn't like my pen anymore. Forgot that I wanted a pen. So here is a nice 5-fold axis of symmetry. It's much harder to see, but there is another 5-fold axis of symmetry right here. So now let's get from one 5-fold axis to the next 5-fold axis. We jump 1, 2, 3, 4, 5. Don't have to change directions at all. So your H or K, doesn't really matter. It's going to be 5. And then your K is 0. So T equals 5 process. There's one other thing that I wanted to mention here, and that is to make these relatively complicated structures, you also require scaffolding proteins. And these are proteins that basically will help you in doing the assembly process. Now, we don't know about SV40, but certainly in the case of something like adenovirus, those are helping to put this whole process together, make that whole structure. So in many cases, the scaffolding proteins, however, are no longer present in the final virion. And that's the sort of definition of a scaffolding protein, is you don't have it then at the end. A couple more examples here really quickly. This is from a bacteriophage, Hong Kong 97. Just the head part. There's also a tail that's on there. One of the really neat things that was found almost 15 years ago now was when they finally got the crystal structure, which apparently took them about 12 years to do, of this capsid. What they found was something really bizarre, and that was that the individual subunits that were making up the icosahedral shell here, and we can look at this. It's a little hard to see, but it's a T equals 7 symmetrical particle. But each of those subunits that was present, and this is just one capsid protein, in the hexameric pieces that all go together, turns out that these capsid proteins were covalently cross-linked to each other. And so they had lysine side chains and acidic side chains, which actually made peptide bonds between the side chains in order to cross-link the individual capsid proteins together. And so this process right here, so this purple one here is cross-linked to this purple one, this green one is cross-linked to this green one, and this light blue one is cross-linked to that light blue one. And what that does is it literally links your proteins together. And this is great because it gives you an incredibly stable structure, because that's not coming apart. Peptide bonds are really strong covalent bonds, really strong interactions. And so what this allows is for this bacteriophage, and it turns out a number of other bacteriophages as well, to have these very thin and very strong shell structures. It's arranged in a perfectly quasi-equivalent T = 7 structure. But yeah, so we can actually take a real quick look at that here. Here is a pentamer right there. Here's another pentamer down here. 1, 2, 1. So a T = 7 particle. It's not just these smaller particles, again, that have quasi-equivalent structures. This is Paramecium Berseria chlorella-virus. It's an algal virus. It's got a pseudo P169 structure, but completely icosahedrally symmetric. And you can actually see the five-fold axes of symmetry here with the gazillion other ones, and I wouldn't ask you to count from one to the next on something like this. It's got a very large genome. It's about a 400 nanometers across. The, until recently anyway, largest virus is the Mimivirus. This is an an amoebal virus, which has a 1,179 T number, 70,740 capsid proteins, but again, arranged in this perfectly quasi-equivalent structure. So this is something which is almost as big as a number of small bacteria, still exactly the same geometrical principles which are being used to put this together. So you can get really big with these icosahedrally symmetric particles. I mentioned helical symmetry. There are a number of viruses that use helical symmetry. Most of these are plant viruses, and then some of the archaeal viruses we'll have a chance to talk about a little bit later on in the class. One of the great things about helical symmetry is that nucleic acids also like to form helices. And so if you're thinking about being a bag that's enclosing nucleic acids, this is a really good way to go. But again, the main problem with helical symmetry is what do you do with the ends here? But you can also define helically symmetrical structures in a similar kind of way, just conceptually, to your icosahedron, where you talk about T numbers, etc. All that this is the pitch of your helix, which is the number of subunits per turn times the displacement. And so if you look at this, how each of these subunits is actually arranged around, in this case, it's tobacco mosaic virus. And the way that this actually works is literally it's a binding of 3 nucleotides per protein, and each of those proteins interacts with each other in an exactly equivalent way as you go all the way along the helix. And if you look at mutants of tobacco mosaic virus that have a bigger genome, it's just a longer particle. Mutants that have a smaller genome gives you a smaller particle. And again, so it's just the pitch will tell you exactly how many of these particles are associated with your genome. You can also do the calculation here. 3 nucleotides per protein subunit, 6,400. Actually, this shouldn't be kilobase pairs. It should be kilobases because it's a single-stranded RNA. You can do the calculations exactly how many protein subunits you actually have in there. So we've got icosahedral symmetry, helical symmetry. Lots of bacteriophage actually combine both of these. This is phage 5KZ, published just a couple of years ago. Has a nice icosahedrally symmetric head structure and then a helically symmetric tail structure. And so quite often, and particularly again in the bacteriophage, we have these combinations of the two kinds of symmetry that are coming together in this process. So that's all the proteins. What about the nucleic acid? We talked about it a little bit when we talked about the helical and helical symmetry, 3 bases per protein that's bound. But how do you go about packaging these genomes? Well, one way is that you have your just packaging relative to the capsid protein, just like you have with TMV. So certain number of nucleotides that each of your proteins bind to. This is, often you can also have the assembly process, kind of like what you're asking about for SV40. Sometimes you'll just assemble around the nucleic acid, and that will give you your structure. It's definitely the case for things like TMV, where you make the genome and then the proteins just stick on top of it. How do you actually get those interactions? Sometimes it's a very nonspecific interaction, positively charged side chains, which should sound really familiar from nucleosomes and histones. Basically exactly the same thing is happening there, just charge-charge interactions. So nucleic acid is negatively charged, positively charged protein. That way you can have your interactions. Often this is now really nonspecific. Many viruses, however, have what are called packaging sequences in them, which is where you have a specific interaction between either your major capsid protein or a different protein that causes that genome and only that genome to get packaged. This is really important if you're a virus which is replicating by budding. You're not killing the cell. There's clearly a lot of other nucleic acid which is present inside your host cell. You just want to package the viral nucleic acid. You don't want to be packaging any random nucleic acid. So having a specific way of packaging your genome is really quite important. A groove in the protein, this actually goes back to what we know from tobacco mosaic virus. You may have seen the individual subunits actually kind of look a little bit like shoes. And it's in where your foot would go in the shoe is the groove that the nucleic acid actually goes into. And so particularly for a lot of these helical processes, there's a groove in there. In some cases, there's actually an active mechanism that packages the nucleic acid inside the capsid. This is best known for the bacteriophage. In fact, for bacteriophage T4, I'll try and bring the image before. Some of you may have seen the lysed bacteriophage with all the DNA coming out of it. The bacteriophage particle in the middle and the DNA just being way huger than the rest of it. Turns out that the pressure that's generated by packaging the DNA inside of some of these structures is really pretty high. It's quite impressive how well it gets in there. So genome packaging, to get that kind of pressure, you actually need to expend a lot of energy. So these are the packaging proteins that are literally involved in pushing the genome in many cases. And then you also have some scaffolding proteins that are involved there. So far, we've just talked about the naked virus particles. Of course, there's envelope viruses as well. There's an example here, I think, of a nice combination of the two. This is a model that one of my students gave me of a retrovirus. It's supposed to be HIV, but it's actually not HIV, with a nice icosahedral structure inside an envelope. And so here's the lipid envelope. These lipid envelopes are made from host membranes, but all kinds of different host membranes. We'll look at different enveloped viruses that are made from nuclear membrane, that are from ER, that are from the plasma membrane, etc. Once, however, it's gotten some of that membrane, you still have exactly the same problem with an enveloped virus as you have with a naked virus. You've got to find the right cell, and once you find the right cell, get into it. That's why you need membrane proteins. So these are viral, usually known as envelope proteins, because that's what that lipid envelope is on the outside. They're inserted into the membrane either during the virus processing or right at the end of the virus processing. Usually they've got these big extra virion domains with relatively small ones on the inside, also known as the ectodomains, and of course they're membrane proteins. They're tied to the rest of the membrane. Of course, you've got a membrane. It's usually a flat process. Virus particles are relatively small, so they have to bud, and that budding process is really very specific to viruses. How does it happen? Some cases, it just seems to be you have your virion. Very often, again, your inside will have some kind of symmetrical packaging of your nucleic acid. it'll just pick up the viral proteins and the host lipid as it comes out of the cell. Number of viruses, HIV is probably the best known of those, have a matrix protein that actually serves as an intermediate between the capsid here and the envelope. Usually any of these viruses are getting put together at the membrane, or you've made your nucleocapsid that gets exported. Another thing that happens in a number of viruses is you get some extra bits of membrane, sort of, but fatty acids that are put together with the membrane when you get that budding process. This is often a modification of some of the proteins that end up in the virus particle. Matrix is probably the best known of those from HIV, which has a myristylation. It's a fatty acid side group which gets added to the actual protein. So I'll just finish up here talking about the assembly process and the disassembly process. And this gets way back to what we talked about right at the beginning, which is this whole virus capsid stability paradox. It's got to be really stable, but also be able to release the genome as well. So metastable is what we talk about it. So some cases it's really stable, other cases it's really not stable. Many cases that's due to proteolysis, and the proteolysis in polio is cleaving up the capsid protein. Didn't talk about it, but we'll talk about that much more when we talk about these single-stranded RNA viruses later on. Influenza has a fusion protein, also true for HIV-1. Hopefully a lot of you have heard of antiviral drugs, like for HIV, protease inhibitors. What they're doing is they're actually blocking this assembly process. That's what those protease inhibitors are doing. And then there's a blocking protein specifically having to do with disassembly. Bacteriophage usually have these specific release of DNA from their capsids as opposed to a disassembly process which takes place, and we'll look at those. A lot of the build drills or release of pressure, etc. And then plant viruses often undergo these really interesting structural changes to have a really stable state when they're on the outside of the cell, and then a really non-stable genome releasing state after they get inside the cell. In fact, that's the CCMV that I have in all my beginning slides. They're at the end. There's the extracellular form and the intracellular form, and how those form those structural changes. So this is just the review of what we talked about today. You can think about this test question. I was going to have it for clickers, but we didn't get there today. I'll post this on D2L, and so you can think about what the answer is for this.